Hello friends, today I am going to explain about protein estimation by Lowry's method. Lowry's method is done using a spectrophotometer and it is one of the oldest and very reliable method of protein estimation. Now we will see the principle of the analysis. So Lowry's method is a combination of biuret reaction and the reduction of falling CO category reagent. So here there are two steps. In the first step of reaction, protein binds with copper in alkaline medium and produces Cu2 plus ions. In the second step, these Cu2 plus ions catalyzes the oxidation of aromatic amino acid present in the protein, such as tyrosine and tryptophan. And at the same time, the fuel category reagent, that is phosphomolybdenotungstate, which is reduced to heteropolybdenum blue, so that a deep blue color will be generated. So the intensity of blue color is depending upon the quantity of tyrosine and tryptophan present. This blue color can be measured at 630 or 630 to 750 nanometer in UV visible spectroscope. Next is sample preparation. Actually, this Lowry's method can be used for estimation of protein in different biological samples. So sample preparation may vary from each other. So usually extraction of protein will be carried out in a buffer, but the buffer should not have any interaction with the reagents used. PBS buffer can be used. For this, we can take a dried protein sample of 0.5 gram or 500 mg, and it can be grinded well using mortar and pestle using 5 to 10 ml of water or distilled water, and then centrifuge at 800 rpm at 4 degrees centigrade for 15 minutes. Then we can filter the supernatant and it can be used for protein estimation. Now we will see the procedure. So first pipette out 0.2 ml, 0.4 ml, 0.6 ml, 0.8 ml and 1 ml of working standard of BSA into different labeled test tubes. Similarly, pipette out 0.2 ml of the sample in another test tube. You can make triplicate for better reading. And make up the volume up to 1 ml in all the test tubes using distilled water. Then add reagent C 4.5 ml to all test tube including standard and sample test tube. Then mix the content of the test tube by vertexing or shaking the tubes and allow to stand for 10 minutes. Then add 0.5 ml of reagent D rapidly with immediate mixing and incubate at room temperature in the darkness for 30 minutes. After that, you can take the absorbance using UV visible spectroscope at 660 nanometer against blank. Now you can draw a tabular column like this and enter the absorbance from the spectrophotometer, and you can go for a standard graph preparation. So now you can make a standard graph. So that can be done in graph paper as shown in the figure. On x-axis, you can mark the concentration and absorbance on y-axis. Then mark the points, the readings that you are getting in spectrophotometer can be marked in the graph like this and draw a straight line. Then your unknown sample, the protein sample, absorbance is also can be noted like a dotted line in the graph. You can mark like this, and from this graph you can find out the concentration of the given sample. This is one method, or you can plot it in a Excel software, Microsoft Excel software. So for that, you first plot the graph and then add a trend line, and you can get R square value also and equation for straight line also. And gradient, you have to divide the sample reading absorbance divided by the gradient then you will get the concentration of protein in the given sample.